Mapping and measuring proteins on the surfaces of endoplasmic reticulum in cells. Sigma receptors are proteins found on mainly the surface of endoplasmic reticulum in certain cells. Sigma-1 and Sigma-2 are the two main classes of these receptors. The Sigma-1 receptor is involved in neurological disorders and certain types of cancer. To understand better how the receptor is involved in disease and whether drugs developed to target it are working, it is important to be able to accurately trace the Sigma-1 receptor. Researchers at Kanazawa University have developed a probe which can identify and latch onto the Sigma-1 receptor. The research team led by Kazuma Egawa had previously developed molecules with such binding potential. However, upon detailed analysis of the Sigma-1 receptor structure, they realized that extending the length of these molecules would increase their binding affinity further. The team therefore created molecules with varying lengths and found one probe that bound to the receptor exceptionally well. For measuring and mapping the Sigma receptors by nuclear imaging, radiolabeled iodine was introduced into the probe. The structure created subsequently bound to both Sigma-1 and Sigma-2 receptors. Since Sigma-1 and Sigma-2 receptors are involved in prostate cancer, the team then tested the effects of their newly created molecular probe on prostate cancer cells. These cells have Sigma receptors and thus any probe with a high affinity will attach to them and enter the cell. As expected, the probe signal from within the cell was high. When haloperidol, a drug specific to the Sigma-1 receptor, was added to the mix, this signal dropped, suggesting a competition between the two. To finally assess the affinity of the probe for different tissues within the body, mice with prostatic tumors were used. While the probe easily entered and stayed within the tumors, its presence in the muscles and blood was less. The probe was thus highly specific for tissues with the presence of Sigma receptors. This study reports a sophisticated probe that binds to Sigma-1 receptors better than previous probes developed. This, coupled with its ability to escape non-specific tissues, is a promising step forward in studying changes induced in the Sigma-1 receptor in various disorders. The probe can also be used when developing drugs against the Sigma-1 receptor to compare the binding affinities of such drugs. These results provide useful information for developing Sigma-1 receptor imaging probes, conclude the researchers. RNR switch offers hope in battling antibiotic-resistant bacteria. New research from Cornell University offers a new pathway for targeting pathogens in the fight against antibiotic-resistant bacteria. As antibiotic resistance rises, the search for new antibiotic strategies has become imperative. Researchers used the Cornell High-Energy Synchrotron Source (CHES) to reveal an unexpected mechanism of activation and inactivation in the protein ribonucleotide reductase (RNR). The findings were published in Convergent Allostery in Ribonucleotide Reductase in Nature Communications. Understanding the switch that can turn RNR off provides a possible means to shut off the reproduction of harmful bacteria. RNRs take ribonucleotides, the building blocks of RNA, and convert them to deoxyribonucleotides, the building blocks of DNA. In all organisms, the regulation of RNRs involves complex mechanisms. Without these mechanisms, DNA replication becomes error-prone, and dangerous mutations could occur. Without the RNR enzyme, DNA-based life as we know it could not exist, said first author William Thomas, a graduate student in chemistry and chemical biology. If we understand the RNR off switch well enough, we can take advantage of it by developing our own ways to toggle it with new antibiotic drug molecules. This research reveals evolution in action, according to Nozomi Ando, assistant professor of chemistry and the paper's senior author. The lack of the normal regulatory switch mechanism may provide an evolutionary advantage for the bacteria they studied. Usually the increased chance of mutations is a problem for bacteria, but maybe under certain circumstances it's actually advantageous for an organism to mutate and possibly become resistant to an antibiotic or another stressful situation, she said. RNRs are not easy proteins to work with or understand, and the researchers said characterizing them in the traditional way has been challenging. The combination of small-angle X-ray scattering using CHES, crystallography, and cryo-electron microscopy is what made this study possible, Ando said.